Different species of lymphatic filarial parasites. Lymphatic filariasis is a neglected tropical disease caused by several species of parasitic worms known as lymphatic filarial parasites. These parasites are transmitted to humans through the bite of infected mosquitoes. Once inside the human host, they mature into adults and reside in the lymphatic system, causing chronic and debilitating conditions, such as elephantiasis and hydrocele. This essay will explore and distinguish the key features of different species of lymphatic filarial parasites, Wuchereria bancrofti, Brugia malayi, and Brugia timori. 1. Wuchereria bancrofti. Wuchereria bancrofti is the most common and widespread species of lymphatic filarial parasites. It is prevalent in tropical and subtropical regions, including Africa, Southeast Asia, the Indian subcontinent, and parts of South America. Key distinguishing features of W. bancrofti include: A. Morphology. The adult worms are relatively long and slender, measuring approximately 4 to 10 centimeters in length. They have a well-developed cuticle, allowing them to survive in the lymphatic system. B. Microfilariae. The microfilariae of W. bancrofti are nocturnally periodic, which means they are most abundant in the bloodstream during the night. They have a sheathed appearance with a tapered tail and measure about 250 to 300 m in length. C. Vector. The primary vector responsible for transmitting W. bancrofti to humans is the mosquito species Culex quinquefasciatus in urban areas and Anopheles mosquitoes in rural areas. 2. Brugia malayi. Brugia malayi is another important species of lymphatic filarial parasites that is prevalent in Southeast Asia and parts of the South Pacific. It is closely related to W. bancrofti but exhibits some distinguishing characteristics. A. Morphology. Adult B. malayi worms are shorter and stouter than W. bancrofti, measuring about 2 to 4 centimeters in length. They also possess a well-developed cuticle. B. Microfilariae. The microfilariae of B. malayi are nocturnally subperiodic, which means they appear in the bloodstream at irregular intervals throughout the day and night. They have a sheathed appearance with a more rounded tail and measure about 220 to 260 m in length. C. Vector. The main vector for B. malayi transmission is the mosquito species Mansonia in urban and rural areas. 3. Brugia timori. Brugia timori is a species of lymphatic filarial parasites closely related to both W. bancrofti and B. malayi, but it has a more restricted distribution. It is primarily found in certain parts of Indonesia, particularly on the islands of Alor and Timor. Key features of B. timori include: A. Morphology. Adult B. timori worms are similar in size to B. malayi, measuring about 2 to 4 centimeters in length, but they have some distinct anatomical features that help differentiate them. B. Microfilariae. The microfilariae of B. timori are nocturnally periodic, like those of W. bancrofti, with a sheathed appearance and tapered tail. However, they are generally smaller, measuring about 200 to 250 m in length. C. Vector. B. timori is primarily transmitted to humans by the mosquito species Anopheles barbarostris. It is crucial to accurately identify the species of lymphatic filarial parasites present in an area for effective disease control measures. Different species may exhibit varying levels of resistance to certain drugs, respond differently to vector control strategies, and have distinct geographical distributions. Understanding these distinguishing features helps in developing targeted interventions and implementing appropriate preventive measures. In conclusion, lymphatic filariasis is caused by several species of lymphatic filarial parasites, with Wuchereria bancrofti, Brugia malayi, and Brugia timori being the most prominent ones. Each species has unique morphological and biological characteristics, including variations in adult worm size, microfilarial periodicity, and vector preferences. Such distinctions play a crucial role in designing specific control and elimination programs to combat this debilitating disease effectively. By understanding the distinguishing features of these parasites, public health authorities and researchers can